So how do we get from DNA to RNA to proteins? We have to go through two major processes, transcription and translation. Transcription goes from DNA to RNA. Translation goes from RNA to proteins. So how do we do this? Well, we know the DNA can't leave the nucleus. That's not allowed. So we have to get the information, the coding information from DNA out to make proteins in your cytoplasm. And the only way we do that is through the production of something known as mRNA. So M is messenger RNA, and it's literally carrying the message of genetic code information out to the cytoplasm and the rough ER to match up with the ribosomes to build amino acids and then form proteins. So in this sequence, we select particular genes. So you see here they've got some genes labeled for you. We would pick particular genes. Your body would request particular genes to be amplified and turned into proteins. So if we take our DNA, that's our gene, and we're actually going to open up the DNA just a little bit like we do during DNA replication, and go in and we're going to base pair match the mRNA strand. And you see that. C's goes with G, fine. Why does A go to U? What does A normally pair with? A is supposed to pair with T, but if we are making RNA, we have no T's, right? So A pairs with U. And then from there, we're going to use this thing known as a codon. A codon is 3 RNA bases that is going to be specific to 1 amino acid. So we're taking, in this case, starting with 3, 6, 9, 12 bases. And if we have 3 codons, so 12 divided by 3 gives you 4 amino acids. And you see that here. So this codon takes information and helps us to translate it. So it's part of translation into a new language. So what's it translating? It's translating the language of RNA into the language of proteins, specifically amino acids. For each amino acid, okay, so we have 20 amino acids, remember? For each one, there's a codon specific to it. But there are actually 64 possible codons, because you have to think we have A, C, G, and U in our RNA, right? So in the first position of my codon, if I have 3 bases that are my codon, the first one could be A, the second one could be A, and the third one could be A, right? Or the first one could be A, the second one could be C, the second one could be, or the third one could be G, or G, U, A. You could do this, and you actually get 64 different ones till you get them all done. Okay? AUG is one of the most common amino acids, and we'll see it later on. It's something known as the start codon. It starts, this 3-letter sequence starts every single protein made in your body. Every one of them, no exceptions. So this DNA to RNA to our polypeptide or our protein, this idea of transcription, so going from the DNA to the RNA language, and then translation, so a full translation to a new language of these amino acids. We said that we have 3 nucleotides, so the codon, that's going to specify a single amino acid. We actually have 61 codons, so we had said we had 64 possible. We have 61 codons that are actually going to code amino acids. We have 1 codon that we said is our start, but it also codes an amino acid known as methionine. So that counts as one of these. So we have 3 left. They're known as stop codons. They don't code an amino acid. They're just a big stop sign. We have to know when to start and stop these protein productions. The genetic code itself is what we call redundant. There's more than 1 codon for each amino acid. So if you have 20 amino acids and you have 61 amino acid codons, you've got more than 1 that will code an amino acid. It's what we call unambiguous. It means that for any single codon, it will only give you 1 amino acid. There may be more than 1 codon that would give you that same amino acid, but if I have AUG, I can only ever get methionine. I don't have any other choices. That's it. It is what we call nearly universal because I can take the amino acids out of you, put them into another organism. I can take the DNA or mRNA out of another organism, put it into you. You're going to make the same amino acids. I have here that there's no punctuation. It's just like a big run-on sentence. Okay, so don't think about breaks or anything like that in it. This is a codon table. So here's our AUG that I keep mentioning to you with our methionine. So how does this work? Well, the first base pair I said was A. The second base pair is U. And then the third base pair is G. So if I have a codon that is, let's see, let's look at UUA. What amino acid do I code if my codon is UUA? UUA. I code for the amino acid LEU or leucine. You don't need to know the amino acid names. Don't worry about that. If you can read this table that works a lot like an old multiplication table, you're in good shape. Now we mentioned those stop codons. Here they are. So UAA, UAG, and UGA do not code for an amino acid. They just simply say stop and everything falls apart. We'll show you how this works.